Okay, so now is for the practical part of DNA and central dogma, and that's genetic engineering. What can we do with the information that we have? PCR is a way of taking a little piece of DNA and running it through machinery and making millions of copies of that little piece. So if I were to go off the deep end and murder someone and just leave one piece of DNA, one cell behind, they can make that single piece of DNA in that single cell into millions of copies that they can then run for identification purposes. We're going to have enzymes replicate the DNA without the use of living cells. Back in the day, this used to be like water baths and ice baths, and you would actually take your test tube from here and you'd have to like set your timer on your watch, and then later you would take it over into here and then move over here. Now it's all done in this little itty bitty machine that runs it automatically. The DNA is heated to break the hydrogen bonds between the strands, which uncoils the DNA. The primers are then added to start the DNA replication. As the DNA cools, the primers bond to the original DNA, and nucleotides and DNA polymerase are added. Forms new copy of DNA, which can then undergo the process again and again and again. Often, we're not going to run the entire piece of DNA. We're going to use restriction enzymes to cut out targeted pieces. So in the last lecture, you looked at the fact that there were 13 really segments of DNA that distinguish you from your neighbor. They're going to cut out those 13 pieces and then run those through the PCR and then run them through gel electrophoresis. So here it is. We start with our original piece. We heat it to break it apart, and then we add in the primers and cool it to allow for base pairing, and then heat and cool until we have as many copies as we want. We then take these pieces of DNA and we run them in gel electrophoresis. This is like a Jello jiggler. Essentially, it's made of agarose, which is the slimy stuff on brown seaweed. And that Jello jiggler has got pores in it. And obviously, if you're a small piece of DNA, you're going to wiggle through that pore and go all the way to the end. If you're a large piece of DNA, you're not going to get as far. Think about going through a maze, like a corn maze. We just ended Halloween. If you are going through the corn maze by yourself, you are very maneuverable, right? You can get in and around the corners. But if I ask you to go through the corn maze holding hands with 5 of your friends, it's going to be a lot harder. You're not going to get as far. That's the same thing that's happening in our gel for gel electrophoresis. The fragments are going to move from negative to positive as we're running a charge through this Jello jiggler. So what are we going to do with it? DNA profiling is known as DNA fingerprinting. And the reason why we call it DNA fingerprinting is because as lay people, we understand that everyone's fingerprint is different. But if you don't know science, you may not know that everyone's DNA is different as well. And so we just called it DNA fingerprinting so that if you're sitting in a jury box, you would have a better understanding of this. So we're going to compare DNA from different sources without mapping the entire genome. I don't care what the code is. I just care how big the segment is. We're going to do it for paternity cases, that whole Maury thing. It's based on the concept that a child's DNA comes from your parents, and that a child's DNA, the strands, will either match up with mom or with dad. And so if we have mom here, the kid here, and then here are the 2 suspected fathers, well, this first band on the kid matches mom. Second band, father 2. This band matches mom. These match father 2, father 2. And so you can see that it has to be dad number 2, not dad number 1, because the child matches up with dad number 2. We're going to do it for forensics to help identify criminals and to look at corpses. So if we find a body in the woods, we're going to use the DNA and compare it to relatives. So here we go. We've got a blood stain that was found at the scene, and here are our suspects. So who did it? Match up the blood stain with your suspect. So suspect number 3 matches. Suspect number 3 did it. Again, we've got the blood on the defendant's clothes, and here's the victim's blood, and you can see the victim's blood was found on the defendant's clothes. By the way, doesn't mean that he did it. It just means that he was present and that he got the victim's blood on him. There has to be more to the trial than just the DNA evidence. We have sequenced the entire human genome. It took a lot less time than we thought it was going to, but we did it because it helped us improve our understanding of many genetic disorders. Because of this, we can now screen for a genetic disorder. We can screen to see if you carried the gene for breast cancer. We can screen to see if you carry the gene for Tay-Sachs or any of these other genetic disorders. The downside is that an insurance company could possibly use this to deny coverage. If we know that you've got a genetic disorder like dementia that is going to be late onset, then maybe your insurance company drops you before you get to the average age of onset. In California, they're not legally allowed to do that, but remember, there are ways around that. So instead of saying, I'm dropping you because you've got this predetermined genetic disease, I'm dropping you because you were 12 hours late on your payment. We can produce medicines to cure diseases or genetically engineer to remove a gene that causes a disease. We can now research and have our medical development be gene specific, and we can provide evolutionary pathways. A lot of our animals are getting redesignated in classification because originally we said if it walks like a duck, quacks like a duck, acts like a duck, it must be related to a duck. And now we're finding a lot more convergent evolution, that these two things that we used to classify together are really not related evolutionarily. Watch any of these hyperlinked videos, by the way, you really should. But there was a case a few years ago where the company that discovered the BRCA gene, the one that kind of predetermines you for breast cancer and ovarian cancer, they wanted to patent the technology to isolate that gene, which means anyone who wanted to go for that genetic test, the money would go to that company, and it would be very expensive. The Supreme Court heard the case, and the argument against stated that genes were not created by the company and therefore could not be patented. The argument for stated that that's true, the gene wasn't created by the company, but the technology to isolate the gene was. The final ruling is that you cannot patent a human genome sequence. Note that the genetic code is universal, so it doesn't matter whether you're a mouse, a bacteria, or a human, AUG is the start codon. This allows us to take genes from one organism and put them into another organism. Human insulin is now produced in bacteria. So instead of producing it and isolating it from, say, pigs or humans, we can now produce it in bacteria. Bacteria reproduce very quickly in high numbers. They're easy to keep. It allows us to produce human insulin at a lot lower cost. This whole idea of gene transfer, so vectors carry the gene into a host cell. The host cell is often a bacteria, again, easy to keep, reproduce quickly, can make this gene copies very quickly. The new gene is spliced into a plasmid, transferred to the bacteria cell. The bacteria is then cultured. Restriction enzymes then cut out the desired DNA, and you end up with an uneven cut of DNA, and DNA doesn't like to be cut uneven, so it's got sticky ends, and those sticky ends allow us to put it into a host cell. So in China, they actually took the gene for bioluminescence out of a jellyfish and were able to splice it into a pig embryo. And so now our pig glows in the dark. Here are other pictures of us doing it. BT corn is the IB approved example for this. BT corn was really big because it allowed us to make a corn that was resistant to insects. So now all of our corn crop makes it into market, as opposed to losing a portion of your corn crop every year to insects. Other things that we have genetically modified are GMOs. Were the Flavor Saver tomato. We modified it to stay fresh in your fridge longer so there'd be less food waste. Unfortunately, it didn't taste well, so it didn't last long. We did the BT corn, and we've done golden rice. Golden rice, in some countries where rice is the main source of carbohydrates, there was also a vitamin A deficiency, and if you're deficient in vitamin A, it leads to blindness. And so we basically cut in a gene so that the rice would make vitamin A to hopefully cure that deficiency. GMO, genetically modified organism, GM, genetically modified, or transgenic, all mean the same thing. So if you see it on a test, it's all similar. So BT corn, again, this is your IB approved example. The benefit, Damages caused by insects greatly reduced. Less expensive than spraying pesticides. You have less manpower. You're not exposing humans to pesticides. The harm is that it's going to kill insects, right? So insects are going to lose their food source, and exposure to this gene possibly could also harm the organism if it was able to eat it and survive. And we think that monarchs are a big victim of this. The insects themselves are becoming resistant, and therefore you're still going to have to spray pesticides. And we can't control the spread of genetically modified pollen, right? So if you're trying to be organic and non-GMO in your cornfield, but your neighbor's got the BT corn, when the wind blows, the pollen from his corn is pollinating yours. And so there is this problem that we can't contain this genetic modification. We can clone things now, which is pretty cool. So reproductive cloning creates a new individual, identical genetically to the original. So we take the nucleus from a body cell of the thing we want to clone. We take an egg and get rid of the nucleus and insert the nucleus from the body cell of the thing we want to clone. Then we provide an electric shock, which causes it to start reproducing. And we then implant this embryo into a host. So I love my dog. If you haven't been around me long, you know that my dog is my favorite child. I could conceivably clone him, right? He's a Rhodesian. He's got a very short lifespan. He's almost 5 now. Typically, they live to 9 at the oldest. So I could take a body cell from my dog and send it to China. They will clone it for me and send me back the puppy. But is it really my dog? So a lot of what I love about my dog is from my experiences with my dog, from how I've treated my dog and what my dog has been around and how my dog has learned. So this new puppy would be genetically identical to my dog, but its behaviors, which are not necessarily genetically based, would not be the same. So just something to consider. We can do stem cell research, and in fact, in California at the election, we passed a bond which is going to allow monies to go to stem cells. Embryos are produced and allowed to grow into a small ball of unspecialized cells. At this point, these cells don't know what they want to be when they grow up, and they can be anything. And so then we can, by proximity, have them grow into nerve cells and bone marrow and eye cells and heart cells. And so we can use them to cure a lot of diseases. The ethical issues, in favor of cloning, we could cure disease. Against cloning is the idea of reproductive cloning, of creating and destroying human embryos. This is all stem cell issues here. There are a couple of videos for you to watch at the end of this. This de-extinction video is kind of cool if you're like, I really love bio and I love genetics, but I don't know what I could do with it. The field of de-extinction, like Jurassic Park type stuff, is becoming a big field in our universities. So that's a great video to watch. That's it for genetic engineering.